I put a little note on the board here. If Melissa is here, is Melissa here? Well, if she comes in, I hope she'll see me briefly again. Okay, just to remind you, we'll have our review this afternoon at 5 o'clock in the 6th floor lecture hall. Hope some of you can make it. I think they're useful. Now, this morning, I want to talk to you about, I guess, what you might call the coin of the realm of the nervous system, that is to say, the synapse. The little handout sheet is useful in the sense that it covers the main topics as well as showing you a number of the terms that are used when we talk about the synapse. The way I want to handle it today is to show you three pictures, three different views of the synapse as seen with different methodologies, then talk very briefly about the history, how our knowledge of the synapse developed, and then a bit about the synapse itself. Obviously, this is something that we could spend just months on. Many of you are knowledgeable about it already, but it's always good to see it again. So let's start just briefly with the pictures of synapses in action. Now, believe it or not, this is, by the way, is a typical old reduced silver stain, seen under the microscope at about 1,000 magnification, which was about all you could get with the light microscope. And our best views of the synapse with this kind of technique, believe it or not, was right here. Here you see a fiber that broadens out into a little synaptic bouton that's applied to the surface of the cell body. You can sort of, if you have a good guesswork, conceptual brain, you can think you can see some flattened boutons here, and you can see one making contact there. And that basically, with one or two exceptions, is the best we could do with the light microscope. So it was orders of magnitude different when we could go to the transmission electron microphotograph, go up to powers of, this may be about 60 or 80,000, and then actually see a synaptic terminal. Here's a terminal or bouton. We can see the synaptic interfaces here, the presynaptic and postsynaptic membranes. We can see the vesicles. We can see another one here. Isn't this nice? Nature even put an arrow on this, so we wouldn't miss it. But look at the way the vesicles are lined up and docked along that presynaptic membrane. So this gives you just a totally different idea of the internal anatomy of the synapse. And then from another point of view, here we're using a different kind of electron microscopy. This is not transmission electron microscopy. This is what we call scanning electron microscopy. Its beauty is that instead of looking at very, very, very thin sections, we're looking actually at whole tissue clumps. And the microscopy gives us a three-dimensional version. So what do we see? We see the cell body, the beginnings of dendrites, and we actually see the boutons as little nubbins you could just put your hand around. Isn't that dramatic? And sometimes you can see little boutons on the boutons, and we know that that indeed exists. And you can see some of the little presynaptic fiber that terminates in the bouton. The same here. So with techniques like this, we get a very different idea of what the nervous system looks like. Let's see, I think there's one more, a little more magnified. Yeah. Here we're looking at the cell body surface, a tiny bit of the surface, and these monsters are individual synaptic boutons of the order of a micron in size. So this shows you what modern technology will do. Chris, if you can do it, we can turn off the machine now and save the light. So let's now go and talk about the synapse. Now, the very first line I had down on your handout sheet is an important line. It says, what is the synapse? It's a functional, polarized, which means preferentially it conducts in one direction only, polarized, contiguous, and that's an important word, point between two elements. Contiguous means close but not touching. And that, of course, gets us into the first great fight in what we might call modern neuro, which was the fight between people who thought that in the mature nervous system, every neuron was distinct and made contact with its next friend, but a contiguous rather than a continuous contact. If it was continuous, the cytoplasm would flow from one into the other and make up just an endless network, a reticulum. And they indeed were called the reticularists. The neuronists were those who felt that every cell and its processes were separate entities and were close to and communicated with adjacent ones, but the cytoplasm didn't flow. I can't tell you how bitter that was. I put down the names of Cahal and Golgi. Cahal, of course, was one of the great proponents of the neuron theory, which we all are today. Golgi, using the same kind of methodologies, the Silver-Golgi technique, which they both were masters at, Golgi was a proponent of the reticularist idea, which is strange. Now, the interesting story is that in 1906, Cahal and Golgi were both given the Nobel Prize. They both went up to Stockholm, and they both gave their talks. Golgi gave beautiful material, Golgi stains and so forth, proving that it had to be reticularist. Cahal followed him and gave the same kind of stains, showed how they had to be neuronal in nature. And the two men never talked to each other again. So in other words, there is a lot of feeling in neuroscience. This was one of the early examples. Another great struggle, and these struggles always generate as much heat as light. And the funny thing is, when you get right down to it, that both sides will prove to have some right on their side. Did information carry from one element to another strictly by the flow of electrical currents, or by the release of a small amount of chemical at one of these gaps, one of these clefts between the two contiguous elements? Electrical transmission versus chemical transmission. And by the way, that fight was still going on when I got into neuroscience. So, it's only a couple generations away. The folks who were in favor of electrical transmission only were known as the sparks. And the fellows who were in favor of chemical transmission were known as the soups. And the soups and the sparks had their regular fights at every meeting. And then there were two names that I think were closely associated with these 2 points of view. The first one there, Otto Loewi, who, by the way, as a very, very old man, died relatively recently. Loewi had a wonderful experiment that, for him, convinced him that the chemical idea was the right idea. What he essentially did was, he had a couple of frogs. We'll assume that's a frog. And he had another frog. He opened up into the hearts of these two animals. Here's the frog heart 1, here's frog heart 2. Frog heart 2, he cut all the nerve fibers, particularly the vagus fibers, which, as we know, act on the heart, slow the heart. He cut them all here. This one was intact. Then he ran a cannula from this heart, from inside this heart, all the way over into this side. And then what he did was, he stimulated here. Well, this heart slowed down, and then some seconds later, this heart began to slow down too. And he reasoned correctly that there must be some chemical that's being carried in the fluid, the blood flowing from this heart, which is being stimulated, to this heart, which is only getting an effluent from that first heart. And he called that stuff Vegas stuff, or Wasserstoff in Germany. That sounds the same way. And that Wasserstoff was acetylcholine. And acetylcholine was, in a sense, the first known neurotransmitter. It also turned out to be the transmitter, the neurotransmitter, in all of our neuromuscular synaptic articulations. So as I say, acetylcholine was the paradigm chemical. Loewe at least had made his point out here in the peripheral nervous system. It took John Eccles, again working in the 50s, so I remember it again well. John Eccles showed, using intracellular recording, that on the basis of the results he was getting, synaptic activity could only be working if a small amount of chemical were released at the interface. So these experiments all threw everything very, very heavily in favor of what we came to call the chemical or chemogenic synapse. Chemogenic synapse. What is a chemogenic synapse? It's obviously a synapse that is provided with a chemical mediator that crosses the cleft. Where are those mediators kept? They're obviously kept, for the most part, in vesicles that cluster here. And when a signal comes down, we'll talk about this in more detail later. This is just a sort of an overview. Vesicles will be released, cross that cleft we talked about, and produce some kind of effect postsynaptically, which will be the signal. Now, the chemogenic synapse and the chemical theory seem to have won the day. But as I told you, when you have bitter intellectual fights, it'll usually turn out that there's some justice on both sides. We began to realize that in non-mammalian, in more primitive vertebrate systems, and also to some extent in invertebrates also, there were indeed synapses that appeared to have very, very little cleft or distance between the two elements, and that furthermore appeared to work strictly electrically, without the intervention of a chemical mediator substance. One of the best known examples of that was in the fish brain. Now, in the brain stem of the fish brain, there's a giant neuron, a very big one on each side. And it sends a very big axon down the spinal cord, down to, I guess you'd say, the lumbosacral region of a fish. No reason why a fish should have a lumbosacral cord. And the motor neurons that it drives are the ones that drive the tail, the posterior, the tail of the fish. And a quick flip of that tail gets the fish out of danger quickly. This giant axon that comes down, called Mouthner's cell, comes down here, don't worry about that, comes down, makes an electrogenic synapse here, and is very important in saving the fish from trouble. The beauty of this electrogenic synapse is the speed and the surety with which it works. Speed and surety are the hallmarks we found of the electrical or electrogenic synapse. Then, to our surprise, we began to find these very, very, very close appositions that apparently could drive each other electrically. We began to find that in the mammalian nervous system. make a long story short, today we know that we have both chemogenic and electrogenic synapses throughout the nervous system. I'll tell you a little bit about them, and as we begin now to go into detail. Question?
So just to clarify, Louis is the one that advocates chemical?
Yes, right.
And
Eccles, his work showed that there had to be a chemical mediator, at least in the synapses that he was working with. Right, right. So as I say, both turn out to have validity. Now let's go on just a little farther in our descriptive mode before we go down into the mechanics of things. Notice that on your sheet I point out that there are a number of different kinds of synapses. And we can name them on the basis of what the pre- and postsynaptic components are. Obviously, an axodendritic synapse would be one where a presynaptic axon terminates on a dendrite. If you have an axosomatic synapse, you know what that means. You can have an axospinodendritic, which simply means that on the spine, looking like the lollipops of the dendrite, you have your presynaptic terminal there. These, by the way, are usually facilitatory and are very labile, so they're very interesting in terms of learning theory. And to our horror, we found that almost anything can synapse on anything else. You know what, in a sense, was the final straw that broke the camel's back? We found there could be somatosomatic synapses. You can have a nice cell body over here with its dendrites and so forth, and a nice cell body over here with its dendrites. And if you look very carefully, this one has a bunch of synaptic vesicles here, a presynaptic and a postsynaptic membrane. It looks like they're doing a little dance with each other. So synaptic articulation can apparently occur between almost all elements, all neural elements. Now we now know, by the way, that very close articulations between glial cells are also possible, and indeed may provide a whole new signaling system that we're just beginning to learn about now. So in any case, a number of different kinds of synapses depending on which elements are synapsing with which. Then notice that I mentioned on your sheet divergent and convergent. And this simply means that the presynaptic components may spread very widely to affect a number of elements, and that's indeed what they usually do, something like this. So here's a single cell that's affecting a single axon that's affecting 4 cells. That would be, of course, a divergent pattern. But by the same token, each of these cells here, we know, are receiving many converging presynaptic components right there. So both of these are part and parcel of the connective patterns in the nervous system. Now, when we first began to use the electron microscope, it was found by Gray in England that one could quite clearly recognize 2 general patterns in chemical or chemogenic synapses, and they were accordingly come to be known as Gray 1 and Gray type 2 synapses. What was the difference? Well, the Gray synapse number 1 was characterized by asymmetry in the thicknesses of pre- and postsynaptic components. In the Gray type 1, the postsynaptic tended to be a good deal thicker than the presynaptic. We're going to again talk about what all the structures are in a moment. And another element in the gray type 1 synapse, which is quite consistent, is when you look at the vesicles, these little reservoirs of neurotransmitter, the vesicles were spherical. So we might say that gray type 1 synapses were characterized by spherical vesicles, there was some argument about that, and enhanced thickness of the postsynaptic membrane. But the exciting thing is that virtually all gray type 1 synapses are excitatory or facilitatory. So here we have what has held up pretty well, a nice correlation between some electron microscopic anatomical detail and a physiological correlate. What about the gray type 2? Same general pattern, except that the synaptic cleft is usually a bit narrower. Here, the presynaptic and postsynaptic membranes are quite symmetric in thickness. We call it a symmetrical synapse. But the vesicles, and as I say, there was on and on some discussion about this, it's pretty well involved. The vesicle pattern in the gray type 2 synapse tended to be elongated or elliptical, like that. See the difference? So this was 2, this was 1, and was there a physiological correlate to this? Yes, it turned out that the gray type 2 synapse was invariably associated with an inhibitory type of synaptic response. So, while this is, you might say, more of historic significance, it still is one of the ways in which we label our synapses, and I did want you to be familiar with it. Now, let's talk a bit more seriously about the kinds of synapses from a microanatomical point of view. I've already mentioned the chemogenic synapse, obviously, and we know what characterizes that. The electrogenic synapse, also known as the gap junction, is characterized primarily by a very, very narrow synaptic cleft. Now not very athletic, is it? In the synaptic cleft of the typical chemogenic synapse, the cleft itself is usually 30 to 40 nanometers wide. The synaptic cleft of the electrogenic synapse is usually of the order of 2 to 3 nanometers. We call this a gap junction also. So the term gap junction and electrogenic synapse are synonymous. Now remember, I told you that where there is bitter fighting in an intellectual field, in the long run both sides begin to have some basis of reality, except it takes some time to realize it. How does that apply here? Well, it applies in the following way. We've been almost religious in our pointing out the contiguous aspects rather than the continuous aspects of synaptic structure. However, in the gap junction, as the gap junctions mature, we now know, using the electron microscope, that there are little, believe it or not, protoplasmic bridges that allow actual flow, at least potentially, of cytoplasm from one side to the other. More important, these bridges allow ions to flow from, say, the presynaptic directly through to the postsynaptic. And so if that doesn't look like a reticularist version of the nervous system, I don't know what does. So you see, Golgi and Cajal did not really have to fall out at that time. If they had a little diplomatic understanding, there would have been a kind of neuroscientific UN. It's not doing too well right now, is it? But we might have done better. So I do want you to remember then these two kinds of synapses. Now there actually are other kinds of synapses. The one that I mentioned here for you is the so-called, not a very good term, gaseous transmission type of synapse. And this has been an eye-opener. And we came to realize, and part of the work was done right here, which of course got a Nobel Prize for Dr. Louis Gnarro, the fact that nitric oxide could serve as a neurotransmitter, often working back on the presynaptic element, but not always. And then, even more difficult to understand, a toxic gas like carbon monoxide could also serve under some conditions and in some synapses as a neurotransmitter. So it gives you some idea of the breadth of possibilities that are involved in transmission. Let's talk about the parts of the synapse. Again, most of you know this, but I think we ought to go over it. You're familiar with the presynaptic membrane, which is a highly specialized extension of the membrane. If we look at higher power, we usually find that it's characterized by little hills and valleys. The little hills are known as dense projections. It's on your sheet. Dense projections. And the assumption is that these, almost like little fingers, are there to facilitate the positioning of vesicles. When they're activated, and we'll say how in a few moments, that they get into these little docking positions like that. And then in that position, they can fuse with the presynaptic membrane and then be free to discharge their contents across the synaptic cleft. So that's the presynaptic membrane. Now, on the other side of the cleft is, of course, the postsynaptic membrane. And this, we know, can have many, many different characteristics. Again, of course, it's the classic phospholipid neuromembrane in which are floating all those fascinating molecular chunks that we call ion channels, receptors, transporters. They're all in there and making this ordinarily hydrophobic lipoprotein membrane and make it semi-permeable. Very exciting. Synaptic vesicles, we all know about them. They are invariably characteristic of the presynaptic terminal. And just about everybody agrees that the synaptic vesicle is the repository of synaptic transmitter or modulator materials. There are probably anywhere from 70 to 90 identified synaptic transmitters and modulators. We'll talk about a few of them. Now, one of the very interesting things about synaptic transmitters and modulators is the fact that anti-intuitively, this is very anti-intuitive, there are usually more than one of these transmitters and modulators per presynaptic element. Back in the first half of the 20th century, a great English neuropharmacologist, Henry Dale, just for the fun of it, have anybody here heard of Sir Henry Dale? That shows what fame is. Henry, I didn't know him, but I'll call him Henry. Nice fellow. Henry came to the conclusion that each neuron produced only one neurotransmitter, and furthermore, that if you followed the axon out to its extremities, you would have that neurotransmitter and only that neurotransmitter in each of its terminals. Obviously, if the cell only produces one, that's all you would expect to find. And that was known as Henry Dale's thesis, and it was in all the books, and we all learned it and so forth. I didn't feel that convinced by it, but I have no data. What we know today is that cells produce a number of different synaptic transmitters and modulators, and that if you go into any presynaptic terminal, you'll find 2 or 3 or 4. And that phenomenon, and it's a very important concept, the phenomenon is known as co-localization. Why is it so exciting? Well, it may include co-localized in this presynaptic terminal we're thinking about. You may have a fast-acting, usually facilitatory transmitter, such as glutamate. But you may also have a slow-acting neuropeptide, like neuropeptide Y or enkephalin or something, and 1 or 2 others. Now these are released preferentially, depending on the pattern coming down into the presynaptic terminal. For instance, here is your presynaptic terminal. Let's say that pattern, 2 spikes, comes down. It comes, it's gone in just a couple of milliseconds. And what it apparently releases under these conditions is an equally fast-acting, on-off type of element, glutamate. Now, instead of that, let's assume that the disturbance coming down is like this, projecting over time a ways. What does it do? It may or may not release some glutamate, but in this case, it will release the neuromodulator, say the enkephalin or neuropeptide Y. And that has a slower onset and a much longer effect on the postsynaptic element. All of a sudden, you've developed a kind of a keyboard. From one synapse, you can get very different effects depending on the nature of the transmitter that's released. That is a very important idea. And look, you and I have trillions of synapses, and if each one of those trillions has this range of activities, think how it adds to the richness of synaptic output. I told you before that the beauty of the gap junction, the electrogenic synapse, the beauty of it is that it works very fast, probably 1/10 to maybe 3/10 of a millisecond. And it is very, you can depend on it, it's dependable. Now this fellow here, the chemogenic synapse, works slower, usually somewhere between 0.3 or 0.4 of a millisecond or a millisecond or more. The effects of the released neurotransmitters, say of the enkephalin or neuropeptide Y, may go on for hundreds of milliseconds or seconds or minutes or in some cases even hours. Furthermore, the synapse is not completely dependable. Under some conditions, you may not get transmission across, even though there's some release of neurotransmitter. So here on the one hand, you have electrical certainty. On the other hand, you have chemical, you might say, sleaziness. But I'm all in favor of the sleaziness, because look at the range of possibilities that the chemogenic synapse gives you. So I do want you to remember that. The central nervous system is a noisy system. Statistically, it's more or less dependable, but only statistically. But that's its beauty. Now, having said that, let's continue and talk about the actual mechanism of the synaptic transfer. And again, I realize that most of you are very familiar with this. Presynaptic terminal, postsynaptic terminal, and cleft. Let's put in a few vesicles. An action potential propagating down into the presynaptic terminal depolarizes the terminal, and what it does is it encourages or allows the entrance of calcium ion, not only from the outside, but from areas of storage within the presynaptic terminal. Many people feel that some calcium is stored in mitochondria, possibly in other sites, at the accession of calcium. Now, what calcium does is it releases a cascade effect of molecules. Some of you have heard them. I'm only going to mention them, not even have you put them down, because that's in a sense the work of the courses. But synaptotagmin and synaptobrevin and synaptophysin, the range of protein-like molecules that have been identified and that are involved in the mobilization, the transport, and the eventual docking, that's a nice term, docking of these vesicles is quite remarkable. It's become a little subspecialty in the field of neuroscience all its own. The way vesicles are activated, made to dock, and made to release their chemical. Matter of fact, we have several specialists here in the medical school. Some of you may perhaps know Dr. Schweitzer, a great specialist on this, who's done very important work. So I say this is a little subfield all of its own. With the docking of the vesicle, what happens actually is that the vesicle, let's move it down here, it fuses with the presynaptic membrane like that, and the synaptic transmitter or modulator is released across the cleft. Again, this is something that I'm sure everybody knows. To receive the molecules are populations of receptors, those specialized protein-like molecules floating in the membrane. Now, there are several different kinds of synapses. I'm going to really schematize and make it just very, very hard and fast, and it's a lot more flexible than that. There are several possible ways in which the chemical, the ligand, as we say, can affect the postsynaptic element. The simplest way is where the receptor is actually part of the channel. And the ligand binds to the receptor. The receptor produces steric structural changes in the channel that it's tied to. You have ion transfer, sodium, potassium, maybe calcium, and you develop a postsynaptic effect. Maybe inhibitory, it may be facilitatory. This kind of rapidly acting, direct ligand to receptor channel complex, we call that an ionotropic synapse. Ionotropic synapse, and it is the quickest kind of chemical synapse. Quite characteristic, for instance, of some forms of glutamate synaptic activity, and certainly of many, perhaps most forms of inhibitory activity. And most of you know, I think, that the major so-called inhibitory transmitters are GABA and glycine. So these fellows are usually in the ionotropic, quick-acting compartment. In all other synapses that we know, the release of material across the cleft activates a receptor molecule in the membrane. And the receptor molecule, in turn, releases a new trigger substance, a so-called G protein, guanosine triphosphate, don't worry about it, just remember G protein. And then the G protein itself may either work directly on an adjacent channel or in a more complicated cascade may work on a number of kinases which eventually work on adjacent channels. But the beauty of this more indirect and longer time-lapsed phenomenon is that this may also work eventually on the genome of the postsynaptic cell, which means that you may get synthesis of protein. It means you may get changes in structure of the postsynaptic element, and by golly, we're getting very close to something called learning changes. So the slower acting G protein mediated synaptic interaction, especially the long one that involves cascades and eventual activation of genome. This kind of synapse we call metabotropic. Metabotropic synapses. Longer time span involved, more intermediate chemical systems involved, possibility of permanent changes in the postsynaptic element. So you can see why that would be important. Among the second messengers that may be involved in this more prolonged metabotropic process are things like cyclic AMP, cyclic GNP. Some of you have heard these terms, we're not going to spend any time on them now, but this is all a very important part of the complex mechanisms of synaptology. And just a word about neurotransmitters. I've listed here for you 3 of the more commonly known families of neurotransmitters. Amino acids. We were very much surprised, even shocked, that amino acids, simple amino acids, that serve as neurotransmitters. Glycine, for instance, glutamic acid or glutamate, GABA, gamma-aminobutyric acid. These are amino acids, and they have these varied effects I've told you about. I made a statement before that I want to modify now. I mentioned to you that in general, GABA and glycine are inhibitory neurotransmitters, and that has held up pretty well. I told you that glutamate, which by the way is the most frequently found neurotransmitter in the central nervous system, that glutamate is usually a facilitatory neurotransmitter, but not always. But these neurotransmitters and neuromodulators, the effects that they produce on the postsynaptic terminal are pretty well determined by the receptors to which they bind, rather than their own chemical constitution. So it is the receptor in the postsynaptic membrane which determines in general what the effects of release of the neurotransmitter will be. Please do remember that. For instance, we know that glutamate has several different types of receptors. A neurotransmitter like dopamine may have 6 or 7. Serotonin has many different kinds. And of course, we're beginning to realize the complexities of these as we begin to try to treat people with neurological or psychiatric illnesses. Now, just a word about what happens to the transmitter after it's released. Many of the molecules, of course, are bound to receptors. Many of the molecules are not. In some cases, such as acetylcholine, the excess neurotransmitter is broken down, in this case by acetylcholinesterase, and the fragments may either be washed away or saved. In other words, you have good healthy choline fragments, good healthy acetate fragments. No reason why we shouldn't use them if we can. In other cases, such, let's just take one, norepinephrine, the molecule is not broken down, but to the extent that it can be, that it can be captured, you might say, it can be brought back up into the presynaptic terminal again to be added to the pool. Now, this process of, you might say, resorption, if you will, it's given a general term, endocytosis. And it's a funny process because what happens is the membrane invaginates this way, the molecules somehow enter, and then the membrane leaves the presynaptic membrane and becomes a rather specially designed vesicle that floats in, and you lose some presynaptic membrane here just as you gain some when the vesicle membrane was added to the presynaptic membrane here. So that presynaptic membrane is a vital, growing and decreasing structure all of its own. Now, we now believe that most of the uptake occurs not through this endocytotic process, but actually through specific molecular transporters located right in the presynaptic terminal. So needless to say, it's a fairly complicated process using, we now believe, several mechanisms in this attempt to save unused molecular fragments of the mediator substance. So we've handled the entire field of synaptology in a highly shortened way, pardon the brevity of it all. You either know this or you will get it in other courses, but I just want to outline it for you. The quiz on Tuesday morning, it'll be short answer, true and false, underline, fill in, that kind of thing. Probably 33 questions, so we can do it quickly. I think we'll have it right here in this room at the beginning, so we sort of get the suffering over with, so we can enjoy the lecture together. And we'll have the review then, and I'll have my office hours in a few minutes. And if by this time Melissa has come in, please